Okay, imagine you have to build a house, but with some pretty insane constraints.
Oh, I like this. Go on.
You have 9 months to build it. It has to act as a complete life support system for a tenant who is constantly growing. And the second that tenant moves out, the house just self-destructs.
That is a perfect description of the placenta. It's the only truly disposable organ in human biology. We grow it from scratch, we use it, and then we just discard it.
And yet, for something so critical to the survival of our species, it's still a massive mystery. Scientists call it the black box of pregnancy.
Well, that makes sense. I mean, you can't just go poking around inside a healthy human pregnancy to see what's happening at the cellular level. It's ethically, impossible.
Which means we're basically flying blind on huge issues like preeclampsia or miscarriage. But that might be changing.
It is. Today we're doing a deep dive into a really fascinating paper published in Developmental Cell this year, February 2026. It's titled Comparative Analysis of Rhesus Macaque and Human Placental Organoids Highlights Evolutionary Differences in Placentation.
That is a mouthful. But our mission today is to unpack how scientists built a, quote, living avatar of a primate placenta in a dish.
And we're going to break it down concept by concept to see how this model works and what it can tell us about being human. It's really a brilliant piece of biological detective work.
So let's start at square one, the setup. Why do we even need this model? I mean, we use mice for everything in science. Why not for pregnancy?
Mice are fantastic models for a lot of things, but for the human placenta, they're just terrible.
Terrible. Why?
Because humans have what's called a hemocorial placenta. Let's break that down. Hemo is blood.
Right.
And corial refers to the chorion, which is the fetal membrane. In us, the fetal cells, these things called trophoblasts, they actually invade the mother's tissue and end up bathing directly in her blood.
It's a really aggressive, invasive interface.
Exactly. It's high risk, high reward. Mice just don't do that. Their interface is much more conservative. The layers are different. Studying a mouse placenta is like studying a completely different architectural plan.
So we need a primate. And that brings us to the rhesus macaque.
Right. They're one of our closest evolutionary cousins. Their hormonal cycles, their pregnancy, and crucially, the structure of those little finger-like placental villi. It's all remarkably similar to ours.
So if you want to understand human pregnancy without endangering humans, the rhesus macaque is the gold standard.
But you can't just breed thousands of monkeys for research. It's complex, it's expensive. So the whole goal of this paper was to build the tissue in vitro.
In a dish to create organoids.
And we should probably define that term right now because it's the absolute backbone of this entire study. An organoid is not just cells in a dish.
Right. A traditional cell culture is like what? A pile of bricks scattered on the floor?
That's a great analogy. You've got the materials, but there's no structure. It's flat. It's 2D. An organoid is more like a miniature Lego castle.
It builds itself.
It self-organizes. It has architecture. It creates a three-dimensional structure that mimics not just the cells, but the function of the real organ.
So, section 1, architecting the avatar. How did they actually build this Lego castle? What was the recipe?
They started with the raw material, of course, chorionic villi, taken from rhesus placentas in the mid to late third trimester.
Okay.
They digest this tissue, basically dissolve the biological glue holding it all together to release the stem cells, and then they plate these cells in something called matrigel.
Matrigel, I've heard of this. It's like a biological jelly, right?
Think of it as a scaffold. In our bodies, cells don't just float in empty space, they sit in a complex matrix. Matrigel mimics that. It gives the cells something to grab onto so they can build upwards in 3D.
Now here's a detail from the methods that I found really surprising. To get these monkey cells to grow, the team had to use human growth factors.
Yes, that's because monkey-specific growth factors just aren't really commercially available. It's a niche market. So they fed the cells a cocktail of human EGF, FGF2, and R-spondin.
And the monkey cells just understood the human signals.
Perfectly. Which is amazing, right? It tells us that the basic signaling language, the chemical words for grow and divide, is almost identical between our species.
So they grow these 3D blobs, but how do they prove they're actually miniature placentas and not just, confused clumps of cells? They had to validate them.
Of course. They used 3 key tests. First was the visual test. The rhesus organoids, or RPOs as they call them, they grew these outward-facing villi. They looked structurally identical to the real tissue.
Okay, so they look the part. What was second?
The marker test. This is like checking the ID cards of the cells. They use staining to look for specific proteins. They found syncytin-1 or SDC1, which is the specific marker for the outer layer, the syncytiotrophoblast.
The STB.
Right. And they found integrin alpha-6, or ITGA6, which marks the inner stem cells, the cytotrophoblasts.
So the right cells are in the right places. But the third test is the big one, because looking right isn't enough. They have to actually work.
That's the functional test. A placenta is an endocrine organ. It pumps out hormones. In real monkeys, there's a hormone called macaque chorionic gonadotropin that peaks early in pregnancy.
Okay.
The researchers tested the liquid medium that the organoids were growing in, and they found massive levels of MCG. We're talking over 1,000 nanograms per milliliter, and progesterone too.
Wow. So it's alive, it has the right structure, the right cells, and it's pumping out the right pregnancy hormones.
Precisely. They successfully built a functional biological avatar.
Okay, let's pause for our first recap. We're about 5 minutes in. Here's what we've established.
Lay it on us.
1, the problem. We can't study human placentas in real time, and mice are just too different.
Check.
2, the solution. Scientists created these things called RTOs, rhesus trophoblast organoids.
Check.
3, the recipe. They took monkey stem cells, put them in a 3D matrixol scaffold, and fed them human growth signals. And finally, 4, the proof. The organoids look right, they have the right cellular ID cards, and they do the work, secreting pregnancy hormones.
A solid foundation. But
Yeah.
now that the house is built, we need to find out who's living inside. We need to take a census.
Which brings us to section 2, the census. And this is where the paper gets really high-tech. They used RNA sequencing.
Right.
I like to think of RNA-seq as taking a census of a city. You stop every person, you look at the tools in their pockets, their RNA transcripts, and you figure out what their job is. You have a hammer. You must be a carpenter.
That's a great analogy. But for the placenta, there's a massive technical hurdle. It's what we call the syncytium problem.
The syncytium. It sounds like a villain from a sci-fi movie.
It can feel like one for scientists, but it's actually the hero of pregnancy. The syncytiotrophoblast, or STB, that outer layer we mentioned, it's not made of individual cells.
It isn't.
No, it's one giant continuous layer with millions of nuclei all sharing the same cytoplasm. It is huge, and it's incredibly fragile.
So if you try to run a standard single-cell sequencing analysis where you have to separate individual cells,
The STB just explodes. It shreds. It's too big and delicate. In a normal sequencing run, you might only detect that, say, 2% of your sample is STB, even though we know it makes up the vast majority of the tissue's surface. You're effectively blind to the most important part of the organ.
Okay, so how did this team solve the exploding cell problem?
They changed the resolution. Instead of trying to isolate the whole fragile cell, they used something called snRNA-seq, single nucleus RNA sequencing.
Ah, so they just pop the cells open and count the nuclei instead, the little control centers.
Exactly. The nuclei are much tougher. They survive the process. And by counting the nuclei instead of the cells, their detection of the STB jumped from around 2% to over 15%. Suddenly, this invisible layer was visible.
And once they could see it, they realized something pretty shocking. That outer wall, it's not uniform. It has different departments.
Correct. This is one of the key discoveries. They found 3 distinct types of nuclei within that single giant layer. 3 different job descriptions.
Okay, let's use a memory palace for this. Imagine the outer wall of a castle. Who's actually on the wall? First up, we have STB1. Let's call them the apprentices.
Good name. STB1 nuclei are expressing high levels of a gene called ERVFRD1. And this is fascinating. It's actually an endogenous retrovirus gene.
What? A virus gene?
An ancient virus gene that evolution captured and repurposed. Its job is to help cells fuse together. So the apprentices are the nuclei that are just finishing the fusion process, just joining the giant layer.
Okay, so that's construction. Next up, STB2, the logistics officers.
These nuclei are rich in genes like ARB27A. This gene controls vesicle trafficking, so their job is to package and ship cargo nutrients, hormones, waste across that critical barrier between mother and fetus.
And finally, STB3, the diplomats.
These nuclei express high levels of immune regulation genes, especially FEC receptors. Their job is to communicate directly with the mother's immune system and basically say, hey, we come in peace, please don't attack the fetus.
So that outer layer is multitasking. You've got construction, you've got shipping and logistics, and you have the legal and immune defense department, all in one giant cell.
And we only know this because they were clever enough to use the single nucleus technique.
So how do these cells decide which department to join? This brings us to section 3, the trajectory. The paper uses something called pseudotime analysis.
Right. Think of pseudotime like this. You have a pile of shuffled photographs of a person growing up. Pseudotime analysis is the algorithm that arranges them in order from baby to adult to reconstruct the timeline.
So you can see the path a cell takes from being a stem cell to a specialized worker?
Exactly. And this is where we see humans and monkeys really start to drift apart.
How so?
In humans, the developmental path from a stem cell to that outer syncytium, it looks pretty linear. It's a straight shot. But in these rhesus organoids, the path branches.
A fork in the road?
A distinct fork in the road. Branch A leads to the logistics officers, the STB2 cells. Branch B leads to the diplomats, the STB3s. It suggests the monkey placenta makes a very clear decision about which function to prioritize in different areas.
Whereas the human version might be more integrated, maybe.
That's the implication. Now, in terms of genes, both species turn off a gene called CDH1 as they mature. That's the glue that keeps cells individual. And they both turn on GCM1, which is the master switch for fusion.
But there's a difference.
There is. The rhesus STB turns on a gene called page 4.
Page 4. What's that?
Well, in humans, page 4 is usually a marker for stem cells, the very immature ones. But here, the monkey is using it in the fully mature, functional outer layer. It shows that even when the final structure looks the same, the molecular instruction manual has some very different pages.
Okay, time for our second recap. We're about 10 minutes in.
Let's do it.
First, the tech. They switched to single nucleus sequencing to solve the syncytium problem and finally see those giant outer cells. Second, the census. That outer layer isn't uniform. It has 3 departments: the apprentices doing fusion, the logistics officers for transport, and the diplomats handling the immune system.
Got it.
And third, the trajectory. Human development looks like a straight line, while the monkey path has a fork in the road. And monkeys use different genes, like page 4, in their mature cells.
Which brings us to the most dramatic part of this entire story. Section 4, the invasion.
The invaders. These are the extravillous trophoblasts, the EVTs. These are the cells that actually leave the placenta and burrow deep into the mother's uterus.
And here, the researchers hit a wall. In that standard organoid culture we talked about, there were almost no EVTs. They didn't grow naturally.
So the model was incomplete.
Exactly. So they had to develop an induction protocol. They treated the organoids with a special cocktail containing a growth factor called neuregulin-1, or NRG1.
Neuregulin sounds like something from the nervous system.
Biology loves to reuse its tools, and when they added NRG1, the EVT population just exploded. It went from less than 4% to about 21% of the cells. They had successfully flipped the switch for the invasion program.
And let me guess, they found subtypes of invaders too.
They did. They found EVT2, who we can call the rookies. These are early transitional cells. They're laying down tracks with a protein called fibronectin. And then you have EVT1, the veterans.
The mature invader.
The fully mature ones. They express things like NCAM1 for adhesion, and crucially, MMP2.
MMP2, what's that?
Matrix metalloproteinase 2. Think of them as molecular scissors. These cells use MMP2 to physically cut their way through the mother's tissue.
Okay, so we have the monkey invaders. Now for the million-dollar question, the whole point of the study. Do the monkey invaders look like human invaders?
And this is the climax of the paper. The answer is a huge, resounding no.
How different are we talking?
When they compared the genetic markers, the gene expression profiles of human EVTs and rhesus EVTs, only 6.4% of the key markers were shared.
Wait, say that again, 6.4%. I thought we were something like 93% genetically similar to macaques overall.
We are. But in this specific cell type, the one responsible for invasion evolution has gone down 2 completely different roads. It is by far the most divergent part of the placenta between our species.
Wow. So how do they differ? What's the actual mechanism?
It boils down to how they move and invade. Human EVTs rely heavily on something called small GT-pace signaling.
Okay, what does that mean in simple terms?
GT passes are like molecular switches that control the cell's internal skeleton, its cytoskeleton. So human invaders act like little amoebas or special ops soldiers crawling through mud. They are constantly reshaping their bodies to squeeze between the mother's cells. It's a very active, movement-based invasion.
And the monkeys.
The rhesus EVTs rely more on what the paper calls DNA conformation change.
So we're not crawling, they're what?
They seem to be physically reorganizing the structure of their genome to change their fundamental cell state. So instead of physically crawling, you could say they're genetically shape-shifting.
That's a massive difference. Active crawling versus genetic restructuring. And
Right.
there was a key gene missing in the monkeys too, right?
The very famous one, HLA-G. In humans, HLA-G is the ultimate immune shield. It's a protein on the surface of our EBTs that tells the mother's killer immune cells to stand down. It's totally absent in the rhesus data.
So they must have their own version.
They do. It's called MAMUAG. But it's so different that our tools barely recognize it.
Why does this all matter? Why did we evolve such a different and frankly more aggressive invasion strategy?
This gets to the core of it. It's likely about depth, the concept of deep placentation. Human placentas invade incredibly deeply into the uterus, right into the inner third of the mother's uterine muscle.
And monkeys?
Much more superficial. They stay near the surface.
So that tiny 6.4% genetic overlap, that explains the difference in depth.
Exactly. And it also helps explain the risk. Humans get preeclampsia all the time. Monkeys generally do not. Deep invasion is a high-reward strategy. You get massive blood flow to fuel a giant, energy-hungry baby brain. But it's incredibly high risk. If those crawling soldiers, that GT pace mechanism, don't invade deep enough, the plumbing doesn't get set up right, and you get preeclampsia.
That really is the aha moment.
Yeah.
We always think of an animal model as just a simpler version of us. But what this shows is that the invasion is where humans are fundamentally evolutionarily unique.
That's the takeaway. The paper gives us a roadmap. It says, look, use the rhesus model for hormones and basic structure. It's great, but be very, very careful using it to study invasion or preeclampsia because the fundamental machinery is different.
Let's hit our final recap before we wrap up.
Sounds good.
1, the induction. We can force these organoids to create invader cells, the EVTs, using a chemical called neuregulin-1.
Yep.
2, the shock. The EVTs are the most evolutionarily distinct cells. They only share 6.4% of their key genetic markers between humans and monkeys.
The big one.
3, the mechanism. Humans use movement signaling to invade deep, while monkeys use genetic structural changes to invade shallowly. And 4, the takeaway. This difference is likely why human pregnancy is uniquely prone to diseases like preeclampsia.
Before we finish, we should probably mention the one main limitation they point out.
Go for it.
They did manage to create organoids from the mother's side of the equation, the decidua, but they couldn't get them to differentiate properly yet. We don't know the right recipe for the soil. We only have the recipe for the seed.
So we have the invader, but we can't yet model the terrain it's invading. That's the next frontier.
Exactly. But even so, we've cracked open the black box. We have a tool to test drugs, to study disease in our closest animal model, while also having a precise genetic map of exactly where the critical differences lie.
It's incredible. We've gone from a totally opaque black box to a transparent dish.
It's the power of comparative biology. You learn just as much from the differences as you do from the similarities.
So that leaves us with a final provocative thought for everyone listening. If these invasion cells, the EVTs, are the most different thing about our pregnancies, does that mean a condition like preeclampsia is basically the price we pay for being human?
It strongly suggests that. We made an evolutionary gamble. We traded the safety of a shallow attachment for a deep, high-volume connection, all to fuel our massive brains. Preeclampsia might not be a disease in the traditional sense so much as a side effect of our evolutionary ambition.
That is something to chew on. So here's my challenge to you. Look up the term deep placentation. Think about it. If you could switch off just one gene to make human pregnancy safer, but less human in its invasion, would you do it?
It's a rabbit hole well worth going down.
Thank you for listening to this deep dive into the avatar of the placenta. Keep questioning.
And keep diving deep.